Okay, moving on to the lymphatic system, it's really important to remember that the lymphatic system and the immune system work in many ways hand in hand. We've spent some time talking about the cardiovascular system and how the circulatory system relies on the heart to be able to pump blood. We have arteries that pump blood away from the heart and veins that pump blood or move blood back towards the heart. And we've talked about the differences in the pulmonary and the systemic circuit. Now we're going to add kind of another layer of complexity where the veins are not really the only way that we can return liquid and plasma-like fluids back to the heart. The lymphatic system works hand in hand. It's a little bit different from the veins and the venous system, but it carries out some of the same functions with the addition of allowing a physical space for the immune system to do much of its function. So we're going to start today by talking about the lymphatics and kind of segue into the immune system because the immune system relies on the lymphatic system and the blood system to be able to work properly. It needs that anatomy. And we think of the immune system really as more of a cellular anatomy and maybe a tissue-based anatomy as well as a physiology, more of a physiology aspect. Okay, so just jumping right on in. When we talk about why this matters, you can watch that video on your own time. But obviously, if we're thinking about the immune system, we're thinking about blood return, really a lot of different diseases are going to be very important. Most of the things we deal with in a healthcare type setting are going to be directly relatable to our understanding of how the lymphatic system and eventually how the immune system function. Okay, so the lymphatic system and the lymphoid organs will include a network of lymphatic vessels. We call those the lymphatics. Again, those are somewhat analogous to veins, but with a little bit different function. Then we have the lymph, which is actually the fluid that circulates. So rather than actually being plasma, we'll talk a little bit about what lymph consists of, but lymph is the fluid itself. And then we have lymph nodes. And these are sort of cleansing little areas all along the lymphatic vessels. So there's little tissues, little mini organs, if you will, that exist throughout the lymphatic vessels. And we'll look at those here momentarily. So the lymphoid organs and tissues provide structural basis of the immune system to house those phagocytes, things like macrophages that we've already talked about, as well as dendritic cells and some other cell types that we'll get into in this week and next week's lecture. As well as lymphocytes that obviously get their name because they're part of the lymphatic system. And specifically in the next week, we're really going to spend time talking about the adaptive immune system and what we call B and T lymphocytes, which are a very important development of the advanced immune system of mammals and other animals like humans. So the lymphoid organs and structures include things like the spleen, the thymus, the tonsils, the lymph nodes, and then some other lymphoid tissues. Okay, so as a whole, the lymphatic system, essentially returns interstitial fluid that gets leaked by plasma into the local tissues back towards the heart. So it's a very important mechanism for preventing edema. Remember, edema is buildup of interstitial fluid in the local tissues. So, for example, edema in the fingers or in the hands would be when there's fluid coming in from the capillaries, coming from the arteries into the capillaries, going into the local tissue, but then not returning to the capillaries to go out via the venous circulation. So the lymphatic system would help us drain back some of that fluid towards the heart. The lymphatic vessels circulate about 3 liters of interstitial fluid per day, so they're very important. And when we think about the distribution and structure of the actual lymphatic vessels, it's really a one-way system. So it's not like the arteries and veins. It's where you have kind of this continuous circuit. Once things get into the local tissues, they either have to go back out to the capillaries, to that circuit, or they can go on the one-way street of the lymphatic vessels. Okay, so we have lymphatic capillaries, much like we have blood capillaries, and these are going to be what we call blind-ended vessels that kind of weave their way between tissues and cells and the blood capillaries. Okay, so it's not the exact same tubing, if you will, not the same pipes as the blood, but similar in area and location to some of those blood capillaries. We don't have lymphatic capillaries in the bones, in the teeth, which are a type of bone, or in the bone marrow. So Originally, it was actually thought to be absent from the central nervous system, but actually now we understand a little bit better that it does in fact exist in parts of the meninges to help drain the cerebrospinal fluid, or the CSF. So again, similar to blood capillaries, but it's easier for liquid to move in and out of the lymphatics. It's more permeable to the fluids coming in and out. It can take up much larger molecules and particles than the blood capillaries can. So, for example, proteins, some cells, and cell debris, and that includes things like cancer cells, cells that are traveling that should stay in their local tissue but have gone awry and started trying to migrate into another tissue's area, can actually move via the lymphatics, as well as pathogens. So bacteria and non-cellular viruses can actually move around via the lymphatic vessels much more easily than via the blood. Because remember, the blood capillaries are about 1 cell in width, so it's very hard for cells to move through the capillaries. They pretty much have to move single file. So the lymphatics can be an important route for pathogens or cancer cells to actually travel through the body, which is one reason that the immune system is going to use this anatomy, this ductwork, to be able to do its function. And so you can see here in green, and our book very nicely uses the green color to show you lymphatics compared to the red for arterial blood and the blue for venous blood. So you can see the green, it's going to be coming from this local tissue. That could be anywhere in the body that has local tissue, with the exception, like we said, of bone. And basically, it's going to be an alternative route for liquids in the form of lymphatics to return back towards the heart. And so they're going to return to the heart right at the end of the venous system. Okay, the lymphatic capillaries have increased permeability, and they actually use little mini valves that help to propel things forward. Mini valves are anchored in by collagen filaments. So if you remember from BIO201, collagen filaments are those stronger, kind of like the steel cables of the different types of connective tissue fibers. So they increase the extracellular fluid volume. As you get an increase in extracellular fluid volume, you actually open up the mini valves, and that pressure increases. Decrease in extracellular fluid causes the mini valves to close. So think about kind of like a revolving door, a one-way door that can be pushed open, much like the way we talked about some of the heart valves. That same idea, kind of like a bar room door that pushes one way open and then automatically closes when there's less pressure. So here's kind of a picture of what that looks like. You can see the flap or the mini valve that allows things, when there's more pressure, to open and come into the tissue. It comes in and out. And so the lymphatic vessels give way to larger lymphatic vessels. We often call those collecting lymphatic vessels, so kind of like going from the lymphatic capillaries, equivalent of kind of blood capillaries, going to larger and larger tubings, larger structures. And so they have vessels, trunks, and ducts, much like the terms we use for veins and arteries. The structures here are very similar to the tunics or the layers of the veins, and they have thinner walls with more internal valves. We have a lot more anastomoses. So if you remember from veins, those are those alternative routes, those other connections to get liquid to circulate in the same direction. So we have lymphatic trunks as we get larger and larger. And for this class, I don't require you to know the various regions and how the body drains the lymphatics as it comes back towards the different veins as it goes towards the heart. But there's a lot of details I've left here in the slides if you're curious and want to know a little bit more about how that's organized. The lymph is delivered into 2 lymphatic ducts, the right lymphatic duct and the thoracic duct. The thoracic duct is for most of the body. The right lymphatic duct actually drains essentially the right upper arm and the right side up towards the head and thorax. Each of these lymph are going to, each of these is going to empty the lymph into venous circulation right at the point where the internal jugular and subclavian veins come together on either the left or the right side of the body. So here's kind of a picture of some of that structure. As you can see, things coming together right here. And that's actually where it's going to empty. back into venous circulation. You can see a little better here. And as you can see, we start from smaller structures out in the tissues. They're going to get larger. You can see the lymph nodes, which we'll talk about in a second, that are the kind of green little nodules or bumps through here. We'll talk about their function as it works its way back through towards the heart, towards the venous circulation. So it is possible to have lymphadengitis, which is essentially inflammation of the lymphatic vessels. So you get painful red lines under the skin, and vasovassora can be a problem. It gets congested with blood, and the larger lymphatics, like blood vessels, actually receive their nutrients from the branching vasovassorum. All right, so the lymph itself is a low-pressure system, so think very similar to the veins, and it's propelled essentially by the same mechanisms. You have the skeletal muscles that milk or kind of squeeze upward or downward, depending on their physical location, the lymph towards the heart. Pressure changes in the thorax during breathing can help to move the lymph. It has valves that prevent too much backflow. So like I said, the one-way bar doors that only open one way. And pulsations of nearby arteries can also help to propel the lymph forward. And finally, there are contractions of the smooth muscles within the walls of the lymphatics. With increased physical activity, you get increased flow of lymph. That's important, just like we've talked about with the circulatory system. So the more immobilized someone is, the higher the chances of possible inflammation, and the harder it will be for that local tissue to heal, because it's not able to get rid of the fluids and move those fluids and phagocytic cells that may be traveling through the lymph back towards the venous circulation. All right, lymphedema is localized edema of the lymph, obviously. So we've talked about edema. And this can be caused by anything that prevents normal return of lymph to blood. So things like a tumor, for example, might actually block the lymphatics after cancer surgery or during cancer surgery. So lymphedema may improve if some of the lymphatic pathways are still present. Remember, there's anastomoses, those alternative pathways to get fluid back to the same location. And sometimes those pathways that are left open actually can enlarge to make up the difference. All right, so let's jump right into some of the cells and tissues and specific organs of the lymphatic system. So when we talk about lymphoid cells, we talk primarily of what we call lymphocytes, literally site meaning cell, lymphoid. Okay, we have T cells and B cells, and they're appropriately named based on actually where they mature. Okay, it's important to note that they both originate from the bone marrow, but when we call them T cells and B cells, it has to do with where they mature, the T organ or the B organ, which we'll get into in just a second. So the immune system cells, the T cells and B cells, protect against what we call antigens. So the body develops this very unique, smart way of recognizing physical shapes. And kind of a simplistic way to think about what an antigen is it's simply a physical shape, a piece of chemistry the body can recognize and put some kind of identity with. So think about almost like a snapshot. If you could imagine a surveillance camera taking a snapshot of everybody that walks through an airport or a major mall or somewhere that there would be a lot of foot traffic, and it takes snapshots of people's faces, of a rear view, of a side profile view, of the top of their head from a bird's eye view. It might look at what kind of shoes they wear every day. And so over time, it begins to develop kind of profiles of different physical attributes about an individual. The same kind of idea occurs here with the immune system. We develop the identity of what different antigens look like. Anything the body perceives, in this case, we say as foreign, but the body also recognizes physical antigens or shapes as self. So basically the idea is the immune system needs a way of saying, this particular cell is me, itself, or this particular cell or protein or what have you going through the body is not self, it's foreign. Okay, so antigens exist as both self antigens and what we typically talk about mostly are foreign antigens, things that are physical shapes that we recognize as an attribute of something foreign, very specific. This is what the face looks like of an individual perhaps that's recently been has recently committed a crime, or this is what their fingerprint looks like, or this is what their iris scan looks like. It's a very unique way of saying this is in fact this individual. That's essentially what an antigen does for the body. Is it something that's part of me, it's part of self, or is it something that we know is in fact foreign? Okay, so examples of antigens might be bacteria, parts of the outside of bacteria, the toxins made by the bacteria, or even toxins that just in general get in the bloodstream of some kind of poison, different viruses and their physical shapes, red blood cells that are actually mismatched, as well as different types of cancer cells. So the two cell types that are involved in this are called T cells and B cells. T cells essentially manage the immune response, and some also can attack and destroy the infected cells. And B cells are going to produce plasma cells, which make antibodies. And these essentially are special little tools that mark the antigen for destruction by other parts of the immune system, particularly phagocytes. And if you remember the word phagocytosis, phago means eat, cyto means cell, osis meaning process, or literally the process of cell eating. So antibodies essentially are kind of like the way of flagging an individual for jail, or in this case, flagging a foreign substance for destruction by the phagocyte. All right, there's some other cells that we call lymphoid cells, okay? And so they're important parts of the immune system, and they help with a more generalized response, which we'll talk about in a little bit. And so these are particularly the phagocytes called macrophages, literally large eaters, macrophages. And they're really important for helping to activate the T cells to know what they're supposed to attack, as well as dendritic cells. And they get their name simply because they have large structures that stick out and look almost like the dendrites of neurons. They have no connection between neurons. They just, under a microscope, look like they have dendrites. Okay, so macrophages and dendritic cells, for the purposes of this class, essentially do pretty much the same thing, or very similar things. They help to gobble up and recycle foreign stuff and tell the other cells, the lymphocytes, the B cells and T cells, exactly what is foreign. All right, we also have different support cells of the lymphoid cells, things like reticular cells that exist in the stroma, that tissue around the lymphoid organs. So anytime you see the word stroma in anatomy and physiology, we're talking about like the support cells. In this case, it would be the cells around the immune cells to help provide support and structure for the immune system. So here's kind of a picture, a micrograph showing different types of lymphocytes, like here. You can see the reticular fibers from the reticular cells. Here's actually some reticular cells that are making those fibers to provide that structure and framework for the immune system cells to work. There's a macrophage that could gobble something up because it's a phagocyte. All right, so those are the cell types, the B cells, the T cells, the dendritic cells, and the macrophages. Those are the main cell types we're really going to focus on as we talk about lymphatics and the immune system. And the main function of the overall lymphoid tissue is to basically house and provide areas for lymphocytes to grow and make more and more lymphocytes or proliferate. So they offer a surveillance vantage point. So you can kind of think about in our kind of analogy of checking for potential criminals in a public place. It's almost like they're the areas that are like the surveillance camera rooms where all the security guards sit there and watch and monitor what's going on. So they offer surveillance vantage points for lymphocytes and macrophages as they begin to filter through the lymph. And they're largely composed of reticular connective tissue. So if you remember, that's a type of loose connective tissue, if you think back to BIO201 in Chapter 4. So the macrophages themselves that gobble things up live on those reticular fibers. And the spaces between the fibers offer a place for lymphocytes to exist as they are patrolling the body for potential foreign stuffs. So there are two main types of lymphoid tissues. The first we call diffuse lymphoid tissue, which is kind of a looser arrangement. And that's pretty much in every single organ in the body. Larger collections exist in mucous membranes. And then we also have lymphoid follicles or lymphoid nodules. And so these are these little spherical bumps, if you will, that are tightly packed lymphoid cells and reticular fibers. They have what's called germinal centers, so basically a place that has lots of growing B cells, and may form part of a larger lymphoid organ like a lymph node. Okay, you have small groups of these tissues that we'll talk about in a little bit that exist in specific organs that have a high chance of being exposed to foreign stuff. So like the Peyer's patches and the appendix, which exist in the intestines, where there's lots of natural bacteria to help us break down food that we want to make sure cannot actually enter into the bloodstream and affect other organs. So when we talk about lymphoid organs, we talk about two types. We talk about primary and secondary lymphoid organs. And it's really simple to distinguish the difference. Primary lymphoid organs are areas where we actually mature T cells and B cells, those lymphoid cells, or B and T lymphocytes, where they're actually growing and learning which targets to go attack. Okay, so that is the red bone marrow and the thymus. And I'll go ahead and tell you, we'll mention this again, but B cells get their name because they are educated and mature in the bone marrow, the red bone marrow. T lymphocytes get their name because they mature in the thymus. Okay, so that's an easy way to remember it. B, bone marrow, T, thymus. Kind of a side note, you definitely don't need to know this for the test, but you may see this some in different textbooks and different literature. B cells were originally identified in another species, and there they actually grow in what's called the bursa. So that's where the B comes from. It's just convenient that in humans it comes from the red bone marrow, and bone marrow also starts with a B. All right, so primary lymphoid organs where we actually mature those B and T cells of the immune system. Everything else that's part of the lymphatic system is considered a secondary lymphoid organ. Okay, so that includes the lymph nodes, the spleen, and the mucosa-associated lymphoid tissues. Okay, so that's what we call MALT, which we'll talk about in depth in a second. So here's a picture all over the body. You can see we have lots of different components. Every actual bone that has any kind of red bone marrow, so the epiphyses of the long bones, various flat bones in the body that all have the potential to make, or rather to educate and mature the B cells. So they're all part of the immune system, the lymphatic system. So there's red bone marrow, for example, in the humerus. We have the thymus, that's the other primary lymphoid tissue. Okay, so that sits kind of between the lungs, but still in front of or anterior to the esophagus and the anterior to the trachea. The tonsils, which are around the mouth, we'll talk a little bit more about those. Lymph nodes, which are actually all over the body, they're not actually shown right here. Those are examples of secondary lymphoid organs as well. The spleen, right here on the left side of the body. The Peyer's patches all through the small intestine, and the appendix actually at the beginning of the large intestine. They're right by the cecum. So lymph nodes are the main secondary lymphoid organ of the body. They're all throughout the lymphoid tissue. So hundreds of nodes are found throughout the body, and they're bedded deep inside of mostly connective tissue in little clusters along lymphatic vessels. Some are close to the surface. Okay, so that's things like the inguinal, axillary, and cervical regions of the body where you can actually detect them just below the surface. And this would be where collecting vessels converge into trunks, those larger structures. So I've included this slide again just for reference so you can kind of think about where everything is located. And there are two main functions of the lymph nodes. The first is they actually are basically the cleaners. They act as filters for lymph. So macrophages remove and destroy anything that's a potential microorganism or a debris. Maybe it's an old cell that needs to be recycled that's been circulating through the bloodstream. It's bits and pieces of old cells and junk, cellular junk. Or it's actually something like a bacteria that's been killed. Okay, so it prevents unwanted substances from being delivered back into the blood. The second thing that a lymph node does is it actually activates the immune system. So it's a place where those lymphocytes can become activated if the macrophages and dendritic cells and other phagocytes actually detect a consistent invader as they're recycling and getting rid of debris from the blood. All right, So the basic structure of a lymph node, you don't have to know a whole lot for this test about the basic structure, but as we'll see in many other organs, and if you actually remember back to the nervous system chapters, the central nervous system, we'll talk about that it has two distinct layers. It has a cortex, which is like the outer core, and the medulla, which is in the middle. All right, they're very distinct histologic regions. The whole thing is surrounded by a fibrous capsule, and the fibers from the capsule come in trabeculi, so think kind of like the structure of spongy bone, those trabeculi. So the cortex is that outside, the more superficial area that contains follicles with germinal centers that are a place for B cells to divide. So B cells can grow and make more B cells. Deeper in the cortex, you have an area for T cells to travel through. They're continuously circulating in the blood and the lymph nodes and actually through the lymph. That's why they're in the lymph nodes. And there are a lot of dendritic cells that are in those areas near the T and the B cells both. In the medulla, that inside the deeper tissue, the medullary cords extend inward from the cortex and have B cells and T cells as well as plasma cells, which we'll learn later are mature B cells, making specific antibodies. And so there's a sinus area as well through that. So you can see here you can see the fibrous capsule on the outside. Okay, you can see medulla right here. You can see cortex right here. Okay, and these germinal centers where we're actually going to be making B cells. And you can see we have afferent lymphatic vessels. So lymph is coming in on these afferent vessels and then going out on the efferent side here at what we call the hilum. All right, and here's an actual micrograph showing you a stained lymph node where you can actually see the capsule on the outside. You can see the medulla here, this lighter stained area, and then you can see this outer area, which would actually be the cortex. So what happens, basically lymph comes in via the afferent lymphatic vessels. It's going to exit via the efferent lymphatic vessels, and in between, it's going to go through some sinuses where you have all these different immune cells present. An important note is we can actually have what's called buboes. These are basically inflamed, swollen, tender lymph nodes, and they're basically when the lymph node is overwhelmed with way too much stuff going through. So sometimes it's called swollen glands. Your lymph nodes really aren't glands, but they do have a kind of a swollen structure, and sometimes they get filled with pus as you have lots of stuff flowing through, and the immune system's trying to work through what all is going on in the lymph nodes, what all might be potential foreign invaders. And actually, when we talk about the bubonic plague, so maybe if you've heard of the Black Death, the bubonic plague in particular that affected Europe in the late Middle Ages, that bubonic plague was named after the chief clinical feature of the disease, where you actually get these buboes that form these pus-filled areas of swollen glands. Okay, so when you hear swollen glands, somebody says your glands are swollen, well, actually, it's probably meaning you do possibly have an infection because the lymphatic vessels are leading into the lymph nodes with more buildup, more backup as the body's trying to process any potential infection or infectious agent, things causing the infection. So the lymph node can become a secondary cancer site very often if a cancer, which is basically cells that are no longer listening to the normal cues about when they should die, when they should stop growing. They have all sorts of genetic abnormalities that build up over time. And as some of those cancer cells become a little bit further away from a normal cell, they actually may start to travel and what we call colonize or set up shop and build tissue in another area. So, for example, a lung cancer cell or group of cells might actually try to travel into other nearby tissues and set up more space for them to continue to grow or colonize another space. Very often, whatever that primary site is, that tissue type, those cells first travel via the lymphatic vessels towards a lymph node. So very often a lymph node is the next tissue that the cancer tries to set up and build what we call a metastasis, or a small kind of an organoid, a little piece of tissue growing that's from the primary tissue. So for example, in a lymph, you might have lung cancer tissue growing inside of the lymph, or breast cancer or colon cancer tissue that's traveled into a lymph node and tried to grow and make some very mutant, very abnormal lung tissue or breast tissue or colon tissue. And so cancer-infiltrated lymph actually are swollen but not painful. So that's one way we can actually detect some of the differences, whether lymph problems might be due to cancer or due to some kind of microscopic organism like a bacterial infection. In a bacterial infection, you may get some pain involved. That will not happen with cancer in those lymph nodes as they get swollen. All right, moving on to some of the other organs. We have the spleen. And the spleen is divided into two large sections. We'll talk about this in a second. It's a blood-rich organ about the size of your fist. It's on the left side of the abdominal cavity, just inferior, just below the stomach. It's the largest of the lymphatic organs, or the lymphoid organs, and has its own artery and vein. You probably remember that from the circulatory lab exam. And basically, it's a site of lymphocyte proliferation, so we make more and more immune cells, and some immune surveillance and response. So we're looking to potentially send out more immune cells through the body. It's also a really important place that we cleanse the blood of any old, outdated blood cells and platelets. Basically, macrophages here will remove more debris. So you can see the spleen right there. Here's a picture of the spleen. It's actually been sectioned or cut open here. Here's an actual spleen sitting in the abdominal cavity right there. So there are basically three additional functions of the spleen. It stores and breaks down products of red blood cells. So that would basically be things like iron. If you remember, iron is very important to be able to move oxygen around because of hemoglobin. So we get rid of old red blood cells here and recycle them and their contents, including the iron, so the blood can make more red blood cells as needed. It stores blood platelets and monocytes, which are precursors to macrophages. They can be released as needed into the body. So it's kind of a bank or a sink that stores platelets and stores potential macrophages. And it's a site of production of erythrocytes in a fetus. Okay, so not an adult, but before birth, actually, it's a very important place where we make red blood cells before we start making them in bone marrow. So the spleen is actually surrounded by a fibrous capsule. It has trabeculae, those little fibers that reach their way in. So think kind of like a modified, specialized, very large lymph node. It has some other functions as well, and does not exist on the lymphatic vessels in the same way. And the spleen can be divided into two big areas, the white pulp and the red pulp. Okay? And think just for a second back to blood. We said that the reason that arteries are very red, or I should clarify, systemic arteries and pulmonary veins are very red is because they carry oxygenated blood. And we said oxygenated blood has a very bright scarlet color. The more oxygen in the blood, the brighter red the color with the hemoglobin. Well, that would be the case in the spleen. The section of the spleen that is very red is going to have a lot more red blood cells, and that gives it its color. The section that is white does not. So the white pulp is a site where the immune functions occur. The red pulp is the site where old blood cells and potential bloodborne pathogens actually get broken up and destroyed. So because of so many red blood cells being present with those macrophages, we get the red color. In the white pulp, it's mostly going to be reticular fibers with lymphocytes and a place that we can basically have immunosurveillance going on. Where red pulp, we're breaking down potential pathogens, but also old red blood cells. Okay, so here's kind of a picture. You can see the red pulp right here. You can see the white pulp right here. So you can see the color distinction there very easily. And here it is actually on the tissue. You can see the white pulp in this area, and then the red pulp down in here. And again, it has this blue color because it's been stained. The spleen has a really thin capsule, so if you get a direct pressure blow of some kind, like a sports injury, it's very easy to rupture the spleen. It also sits kind of on the surface. It's more superficial than a lot of the other abdominal organs. So if a spleen is ruptured, usually you end up with a splenectomy, a surgical removal. Um, Now it's becoming less common that we do splenectomies. It used to be automatically when the spleen was ruptured, basically the spleen was taken out to prevent hemorrhage and shock on the body. But now we actually know that the spleen can do a lot to repair itself. So we've actually decreased the numbers of emergency splenectomies, depending on exactly what's going on with the injury. So if the spleen has to be removed, the liver and the bone marrow basically take over its function, both the red and white pulp functions. And in fact, in children younger than 12, the spleen might totally regenerate if there is a small part of the spleen left. So another reason we don't actually fully remove the entire spleen if possible. All right, another organ or kind of group of organs is called MALT, or mucosa-associated lymphoid tissue. So the mucosa, if you remember mucous membranes, if you think back to the integumentary system for a second, we talked about the skin being an important physical barrier between the inside and the outside of the body. Well, the mucosa are very important to help with that barrier in areas that we actually still need things to come in and out of the body. So think like the mouth, the anus, the urogenital tract, the eyeballs, places that we need to still let things come in and out, but need to have some type of protection other than skin. So in these areas in particular, or areas that might be exposed to potential pathogens like bacteria, we're going to have a specialized lymphoid tissue called MALT. Okay, and especially essentially these little tissues are going to help protect us from pathogens that might try to enter the body. And so some specific tissues, we have tonsils, which are going to be near the mouth, the Peyer's patches in the small intestine, and the appendix, which is going to be at the beginning of the large intestine or the colon. So the tonsils are the simplest of lymphoid organs. They're a little ring of lymphatic tissue around the pharynx, and they're kind of a swelling. In other words, they're kind of an enlargement of the mucosa. And we have different names for different tonsils. You don't have to memorize which one is which for this class. But the palatine tonsil, think the palate. Okay, so it's the end of the oral cavity. That's the largest of the tonsils. That's what usually gets infected and very often what might actually have to be removed in surgery. You have the lingual tonsils. Remember, lingua means, think about the word language. Lingua is the Latin word for tongue, so it's at the base of the tongue. The pharyngeal tonsils are in the pharynx area. Typically, we call those the adenoids. So just posterior to the nasopharynx. And then the tubal tonsils. And so those are near the auditory tubes, so closer to the ears. So the tonsils function to basically gather and remove any potential pathogens that might come via food or via air, the main two things passing through the mouth and through the throat down towards either the lungs or the esophagus. They are not fully encapsulated, and they have these little crypts, we call tonsillar crypts, that are basically special areas to trap and destroy potential bacteria or other pathogens. So here's actually showing you kind of the tissue structure. You can see them kind of demonstrated here near the nose, near the mouth. The Peyer's patches are going to be specifically in the small intestine. They're really similar to the tonsils, and basically they function in a similar way. They destroy bacteria to prevent them from actually making it through the intestinal wall. They actually do generate memory lymphocytes that remember potential bacteria and other pathogens that the body might need to destroy in the future if they make it through the intestinal walls. So you can see here kind of the mucous membrane that lines the small intestine. And there's actually Peyer's patches right in here. Here's the smooth muscle layer, and then here's Peyer's patches along in here. They have these like lymphoid nodules that help to protect the small intestine. And if you think about that, that makes sense, because when we get to the digestive tract, we'll talk about the importance of natural flora or natural bacteria that exist in the large intestine especially, but in the small and large intestine help us break up food. So we want to prevent that from getting anywhere else in the body, but still help us break down food that we don't make enzymes to digest ourselves. All right, and so the last part of MALT that we'll specifically name is the appendix. And so it's actually a little hook-shaped organ. If we look at the human model in lab, it's on the back side. We flip out the intestines and look on the back side near the cecum, the first part, the appendix. It's actually not totally useless. It does have a function. For many years, we thought that it was sort of an extra organ that didn't have a purpose, maybe a former stomach. But it actually is really important to assist the immune system. You can survive without it in the case of an appendectomy. So it basically aids in the functions of the immune system, kind of like the Peyer's patches. And it does also have memory. So you can see the appendix right there. So this is the large intestine, and this is the small intestine in the middle. You can see all the Peyer's patches. And then as the small intestine first comes into the large intestine, there's the appendix. And there's the rest of the large intestine. All right, the thymus, we said that's one of the primary immune organs, or primary lymphatic organs, because it actually is important for the maturation of the T cells. So it's found in the neck and extends into the mediastinum, so it kind of sits over the top of the heart. It's where T cells are going to mature. It's most active and it's the largest when you're a kid and as you're going through development. It stops growing at some point in adolescence and then actually begins to atrophy or get smaller as you age. It still produces immunocompetent cells, but much more slowly as you're an adult. And so the thymus can be broken into little lobules, little sections. It does have a cortex and a medulla, so a similar thing we keep seeing. And it makes different types of T cells. We'll get more into that later in the next week. So the thymus differs from other lymphoid organs in a few important ways. The first thing is it has no actual follicles because it doesn't have any B cells. Okay, so those other organs, all the ones we've talked about so far, as well as the bone marrow, actually have B cells. The thymus only deals with maturing T cells. And so because of that, it doesn't itself directly attack antigens or things from foreign substances. Its job is to mature the T cells. So functions strictly in T lymphocyte maturation. It actually has what's called the blood-thymus barrier, and it helps to keep immature T cells that haven't identified what antigen they're specifically supposed to attack. It keeps them from being exposed to other potential antigens in the blood. We have a stroma, which is essentially surrounding cells that help support the thymus. Big take home here, the T cells mature in the thymus, and because of that, we actually don't have any B cells, so the thymus does not directly act in an immune response. It doesn't directly try to combat any kind of foreign substance. Instead, it's there to help mature those T cells so that they can go out in other tissues and have a response. Here's a picture of the structure of the thymus. And keep in mind, we're kind of making broad generalizations. You can understand a lot more detail if you take an immunology class, particularly about how the thymus itself works. So the development of the lymphatic system. By week 5, we actually have a lymphatic system with vessels and nodes starting to form during development. The lymphatic organs, except for the thymus, actually come from the mesoderm. We'll talk later in the semester about those different tissue layers. And the lymphoid organs, except for the thymus, actually develop from the mesenchymal cells. The thymus comes from the endoderm and actually comes out of the pharynx and becomes more specialized. And except for the spleen and tonsils, the lymphoid organs are really not very developed at birth. So you have the spleen, you have the tonsils that have some function, and actually after birth, as you get potentially exposed to pathogens, that's when those tissues are going to finish developing. All right, so that's basically the lymphatic system. At this point, we're going to take a few minutes, and we're going to jump into the beginning of talking about the immune system. Okay, and we'll finish up the immune system next week. So the immune system, why does it matter? Basically, everything in healthcare is dealing with some kind of pathogen or disease. Now, there are some diseases that are autoimmune diseases, which we'll talk about, or that are cancer. They deal with problems related to the immune system. There are some diseases where tissues just no longer function correctly for various reasons, but the majority of things we see in a healthcare setting are because of some kind of pathogen, some kind of foreign invader, or some problem where the body starts to attack itself that's autoimmunity, or the body no longer gets rid of bad cells that aren't functioning properly and instead lets them kind of take over, which is what cancer is. So it's very, very important to have a good general understanding of how the immune system works. So be sure to take a few minutes to look at that slide there on slide 68 about why this matters on your own time. So the immune system, okay? So the immune system provides resistance to all sorts of forms of diseases. And we broadly think about it being divided into two big systems. They work hand in hand and help each other, but there are two different unique systems, okay? One that recognizes general things and keeps general things that are not part of our body out, that we call that the innate or non-specific system. The second recognizes very specific threats that we have faced before and helps us combat them much more quickly and efficiently in the future. And we'll spend a lot more time, particularly next week, talking about that adaptive or specific immune system. Why is it that when you get the chicken pox, you typically don't ever get the chicken pox again in your life? Well, that's because of the adaptive immune system. Going back to kind of our surveillance analogy when we were talking about lymphatics, you can think of the innate immune system as looking for generalized traits, things that would deter criminals, things like surveillance cameras and having that ADT alarm system or your Ring video doorbell, having those security guards around and the metal detectors that would help deter potential threats from making it inside of an airport or inside of a big concert venue. Okay? The adaptive immune system instead would be like having those wanted posters with security guards ready to look at a specific person and recognize from their fingerprints and their picture that it is in fact somebody who's already committed a crime. Okay, so innate, generalized immune system, the adaptive immune system specific to exact infections, exact invaders that are bacterial or viral or some other specific antigen that we recognize. And as we talk about those defense systems, we talk about kind of the body's lines of defense. So if you think about this sort of in a security setting or in kind of a military setting, we have sort of our first defense mechanisms and our second defense mechanisms that get employed next. Both of those are generalized and part of the innate immune system. And then we have a third line of defense that's very specific. And this is important. Very often I use this as a discussion question. Can you explain the differences and give me an example of the first line of defense, the second line of defense, and the third line of defense in our immune system? So the first line of defense are going to be physical barriers, things that separate inside and outside of the body. So namely the skin and the mucous membranes, the mucosae. So we very easily prevent bacteria from coming inside of our hands because it's covered in skin. Pretty basic. The second line of defense are actually going to be other types of barriers such as chemical barriers or certain cellular barriers that generally combat things that might be a problem while not harming our own individual cells, our self cells. Examples would be proteins that might be antimicrobial. Actually, lysozyme, which is produced by the body, is actually much more potent and stronger than bleach or 409 at killing most bacteria. So that's produced by different parts of our body that are potentially exposed to bacteria. We have those phagocytes that we already talked about, the macrophages and the dendritic cells that are surveilling potential debris all throughout the body. And if it sees something that's not a happy, normal, healthy, functioning cell, it's going to gobble it up and try to decide if it's actually an invader to potentially activate the third line of defense. But at minimum, it's going to help just get rid of stuff that's not functioning properly, which would include foreign things. And then as well as inflammation, which we'll talk about momentarily. So all of these, phagocytes, inflammation, and antimicrobial proteins are very important parts of the second line of defense. The third line of defense is going to be actually attacking a specific foreign substance, okay? So this is kind of like the security guard arresting somebody that has a very specific crime history, okay? We know we've seen a Staphylococcus aureus infection before. We know what physical shapes the staph bacteria has. We recognize it, and so we're going to go actually attack it and get rid of it before it causes us any problems. All right? So the immune system is a functional system, like I said earlier, rather than really a true organ system. It relies on the bloodstream. It relies heavily on the lymphatic system. But it also has some components as part of other systems as well. So the innate and adaptive defenses are really intertwined. Okay, so while I said you have a first line and a second line and a third line of defense, the third line of defense actually relies on the cells as part of the second line of defense. So our specific immunity, the adaptive immunity, is actually going to require information and the cooperation of the generalized macrophages and other cells that are part of the second line of defense. Okay, so broadly, this is kind of a nice list of innate defenses versus adaptive defenses. We're going to spend next week talking a lot about the adaptive defenses with B cells and T cells in particular, and different aspects of humoral or blood-based immunity and cellular immunity. Innate defenses, the surface barriers, or internal defenses. So first line, second line, third line. So our innate defenses. We use this as the first and sometimes the second line of defense to stop attacks by pathogens like bacteria. The surface barriers are going to be things like the skin and the mucous membranes, like we already mentioned, as well as their secretions, things that they specifically use to help eliminate bacteria. So it's a physical barrier to most microorganisms, and the keratin of skin is resistant to weak acids and bases like the bacterial enzymes and toxins, so it's hard for them to eat through the skin and find a hole to actually get inside the body. The mucosa actually have similar barrier mechanisms. And we actually have what we call the acid mantle. The skin is a slight acid, as well as many of the mucous membranes have at least an acidic secretion if they themselves are acidic. And this actually helps prevent most bacteria from growing in those areas. So the bacteria cannot make it inside of the body. We also have specific enzymes, like I mentioned, lysozyme of saliva. The respiratory tract has different mucosa, and there's lacrimal fluids within tears that do an excellent job actually breaking down bacteria and preventing them from actually making it inside the body. We have mucin and defensins and other chemicals. Again, same idea. They're going to help us prevent things from making it inside of the body. Specifically in the respiratory system, we have mucosae that have hairs, as well as in the nose there are hairs. The cilia is really a more proper term for the hairs in the upper respiratory tract, but those nasal hairs and cilia of the upper respiratory tract help to actually sweep out dust and potential bacteria that may be built up in the mucus towards the openings, towards the nose, towards the mouth, so we can get rid of them. And surface barriers that actually get breached by some kind of a cut or a little nick may make it inside the body, then they'll face the second line of defense. So here's a nice little chart talking about the first line of defense. So the second line of defense, something actually makes it through the skin or passes the mucosal barriers. And this is where the innate immune system really is going to kick in. And so we have phagocytes. We've already mentioned those briefly, like macrophages and dendritic cells. We have natural killer cells, which we'll talk a little bit more about, as well as cells that are part of we have the inflammatory response. It's going to have some of those same cells as the medicinal cells and chemicals. And then antimicrobial proteins, specifically like complement and interferons. And then finally, fever. Those are all parts of the second line of defense. So many second line cells actually have pattern recognition receptors. So you may hear them called toll-like receptors. And basically, these are specific ways of recognizing something as potentially foreign. They recognize different shapes that are common to bacteria, to fungi, to different foreign substances that the body might regularly see. When they recognize those, they can trigger an immune response. So it's not specific where it's like, oh, we recognize this has caused a problem before. Instead, it's like, well, we recognize that potential bad things may have a gun in their hand, or they may have a bandana over their face, or something that's a generalized feature that means it warrants further investigation and possibly to eliminate the threat. So first we have phagocytes. So we've already mentioned a few, but phagocytes would be any kind of white blood cell, so leukocyte, that ingest or digest foreign invaders. They literally are cell eating. Specifically, neutrophils are going to be the most abundant phagocytes. They actually die in the process of fighting off diseases. They become phagocytic on exposure to some kind of infectious material. And you may remember we talked a little bit about neutrophils before when we talked about the different types of white blood cells. A neutrophil count gets high, causes your white blood cell count to get high when you typically have an infection. Macrophages, we've already mentioned. Monocytes are immature macrophages. They can also become several other cell types, but most of them in the body will actually mature into macrophages, which they themselves actually digest potential cell debris, potential foreign invaders. You have macrophages throughout tissues all over your body. You have some that are free macrophages that just wander throughout different tissues, throughout the blood, gobbling up anything that might be foreign or might be old and need to be recycled. You have others that are actually fixed to a specific tissue. So, for example, in the brain, you have microglia. Okay. In the liver, you have stellate macrophages. You have different names that we assign to those different tissues, resident macrophages, that help to surveil against potential threats. For this class, just understand the idea that macrophages are cell eaters that can go throughout various tissues and throughout the bloodstream and throughout the lymphatics to be able to surveil potential invaders, gobble them up, and recycle them, as well as other debris from our own cells or cells that need to be recycled. So here's actually a picture of a macrophage. It's been pseudo-colored, artificially stained, but you can see the macrophage and some of the green rod-shaped bacteria, the bacilli, that they're getting ready to engulf and basically neutralize and recycle its contents. So phagocytosis. And if you look at the study guide, I don't ask you to know a lot of the details of phagocytosis. Just understand the idea that it is, in fact, cell eating. Okay? So the process starts when typically the phagocyte recognizes a specific sugar or carbohydrate structure. It undergoes what's called opsonization, where the immune system uses antibodies or complement or other systems that we'll talk about momentarily to actually surround the pathogens. And they act kind of like handles for the phagocytes to grab onto and to kind of get the whole process started. Okay, so first the phagocyte actually adheres to whatever pathogen or debris. Then we have different extensions that come out from the cytoplasm and essentially engulf and totally surround the foreign substance. It comes into the cell as a vesicle, which eventually forms as part of the lysosome. If you remember, the lysosome has that really low pH. It has lysozyme enzymes that are very much like a strong acid that basically break everything down. Whatever's left gets recycled out into the cell to be able to be reused. Okay, so something gets totally engulfed inside the cell, like right here. Once it does, it's going to fuse with a lysosome that has those enzymes that are very acidic. It breaks down everything inside of the bacteria or cell debris or whatever it is that's trying to be recycled. And then that toxic stuff is small and it can either be ingested back out or recycled inside the cell. So some pathogens are not killed with that acidic enzyme from the lysozyme. Example would be actually the bacteria that causes tuberculosis. And so it actually can use different types of T cells. We're not going to focus on it here. But basically it can use other parts of the immune system to help kill off those cells. We have another important non-phagocyte that's very important in that second line of defense, and they're called natural killer cells. Basically, they attack self cells that may have problems. It looks for self-antigens that may have some issues. It's particularly effective at killing cancer cells and virus-infected cells. So if you think about it for a second, a virus is not actually a living thing. It's just a bunch of genetic material, either DNA or RNA, that's packaged inside of some kind of protein or sugar or fat that floats around, okay? And it potentially tries to infect and put its DNA or RNA inside of either a bacterial cell or a plant cell or an animal cell or some larger eukaryote, okay? Or prokaryote. So in our case, when we get a viral infection, essentially it's infectious genetic material. Okay, it's not like it's a bacteria that does all the functions of a living thing. So there's nothing that we can actually attack and kill because there's nothing living to kill. So what we actually have to do very often is get rid of cells that are infected, kill them, and recycle them so the body can remake that tissue with normal healthy cells. Similarly, cancer cells are cells that no longer listen to normal cues about when to grow, when to stop growing, when to recycle themselves. So natural killer cells are specialized parts of the immune system that just detect problems, generalized problems with cells and their surfaces that indicate they may actually have cancer or they may be infected with a virus. And so the natural killer cells naturally kill our own cells so they can be recycled. Okay. They also and so those cells would actually recycle themselves via apoptosis. Apoptosis may or may not have been discussed much in the cell biology chapter. Maybe kind of assumed you knew this from other biology courses, but essentially that's cell death that's like recycling or programmed cell death. That's very organized so we can reuse all the contents within the cell. Additionally, natural killer cells will basically start inflammation or help to increase inflammation by some of the chemicals that they produce once they start trying to attack the self cells that have been compromised by cancer or virus. Inflammation itself is triggered whenever the body has some kind of injury. Okay, so it could be due to trauma, it could be extreme heat, it could be some kind of chemical irritant, or the infection of a microorganism. Okay, so inflammation basically helps to prevent the spread of the damaging agents, whether it's a pathogen or a chemical or some kind of physical trauma. It helps to get rid of any cell debris that was created by the trauma and helps to get rid of the pathogens. And it's going to alert the specific part of the immune system, that adaptive immune system, to get involved, learn what the problem is, combat it more specifically rather than just general responses, and remember it for the next time you may have the same potential invader. So basically, inflammation is there to get things going for repair, getting rid of the problem and starting repair. So the four main signs, the cardinal signs, this is important in your study guide. The four cardinal signs of inflammation would be redness, heat, swelling, and pain. Sometimes we also add in there, depending on what tissue it is, if movement or use of the area is hampered. So basically, you can no longer use that tissue like you normally could. The four main signs, though, would be redness, heat, swelling, and pain. Inflammation goes through specific stages as it progresses. First, we release inflammatory chemicals that start the process. Then we undergo vasodilation. So if you remember, vaso meaning blood cells, or blood vessels. Dilation meaning getting bigger, like when we dilate our pupils in our eyes at the eye doctor. So increased blood flow because we're vasodilating, and increased vascular permeability. So in other words, we make it easier for blood to come from the blood vessels and the capillaries into those local tissues to carry in potential parts of the immune system, those immune cells that might help to get rid of the problem and start to heal things. Finally, we will actually mobilize the phagocytes, like the macrophages, like the neutrophils. So the inflammatory chemical release, okay, very often it's something like histamine. And so they're released by the injured tissues or the immune cells nearby. And those triggers, there can be other types of triggers, things like prostaglandins, cytokines, if there's a pathogen that's involved, like a bacteria, as well as complement, which we've mentioned a few times. We'll talk about what exactly complement is in a few minutes. They all will cause the vasodilation of local arterioles, so more blood can make it through. It makes the capillaries more leaky, so it's easier for things to make it into the local tissues. It helps to attract the phagocytes that'll gobble up foreign stuff and recycle them. So vasodilation and increased vascular permeability. Basically, we get hyperemia, so lots of blood in the area. It gets very congested. That causes the redness and the heat that we see as signs of inflammation. And the increased permeability causes an exudate, or a fluid containing various clotting factors and antibodies to actually get into the tissue. That's what causes the swelling or the edema in the local tissue. And it's going to push on nerve endings, which result in the pain because there's lots of fluid in that area moving on the nerves. The pain can also result from lots of toxins that get released if it's a bacterial invader, or because of different chemicals like the prostaglandins. We have the edema, the increased blood fluid, essentially, so it's easier to take the fluid back out to the lymphatic vessels and lymph nodes where we have more immune cells that can recycle them and figure out what the potential invader is to present the problem to the adaptive immune system that recognizes the specific threat. And it also can help deliver more clotting proteins as well as complement factors that help to essentially form a meshwork or a scaffold to repair the area. And we talked about that some, if you remember back to chapters 4 and 5 in BIO201, we talked about, for example, skin repair. So lastly, the phagocytes actually mobilize. You get lots of neutrophils and later macrophages. And inflammation is due to pathogens. We also will activate things like complement and potentially the adaptive immune system with B and T cells. Specifically, to get the phagocytes there, first we make more leukocytes. We call that leukocytosis. So that's going to be more neutrophils coming from the bone marrow. Then we have margination, where actually endothelial cells, so those lining cells of the capillaries, get inflamed. And they put out molecules into the outside of the vessel to essentially act kind of like Velcro or little sticky hands that help to pull in passing neutrophils, so they actually slow down and get pulled out into the local tissue. We call that margination. And then diapedesis, which is actually when neutrophils get really small and squeeze through the little holes between endothelial cells that are the lining of the blood vessels to get into the interstitial spaces of the tissue. Okay, so leukocytosis, we make lots of neutrophils from the bone marrow. They move through circulation, and margination is actually where the lining of the capillaries, those endothelial cells, actually pull in or help to grab onto passing neutrophils. And then diapedesis, where the neutrophils flatten and squeeze through the space. And then finally, chemotaxis, which is essentially chemical attraction of inflammatory chemicals that are going to help pull those neutrophils toward the specific spot of injury. So kind of put it in a time frame. About 12 hours after leaving the bloodstream, monocytes are going to be transformed into macrophages. And the late arrivers actually will replace the neutrophils as they begin to die out and clean up all the stuff that's being repaired. So here's the example of a nail coming through your skin, okay? And so you have leukocytosis, where you get more neutrophils going through the blood, margination, where they actually get attracted to the edge of the capillary wall, diapedesis, where they squeeze through the capillary walls, and then finally where they're going to be attracted by chemotaxis towards the site of the injury. And there, the neutrophils first are going to help do repair, and neutralize any foreign substance, like in this case the little green bacterial rods that are coming in. And then eventually you're going to get macrophages that come in and help to clean up the dying neutrophils as well as the debris left from anything in the area and help to start the repair process. Okay, so here's kind of a summary chart. We've mentioned the word pus, but haven't talked exactly about what that is. Pus is basically a creamy yellow mixture. It can be slightly varied in color, but it's kind of a creamy texture. And it consists of dead neutrophils, different tissue cells, and the living or dead pathogens that might exist from where there's first a breach in the barriers. If it gets bad enough, we call it an abscess, and this is actually where collagen fibers get laid down and basically wall off the area that has pus. Eventually, it will have to actually be surgically drained because we're kind of repairing the connective tissue structure where the fibers are, and actually leaving that little pocket of neutrophils and local tissue cells that are being basically cleaned and attacked by the neutrophils to get rid of any living or dead pathogens. Okay, so an abscess is actually kind of a pocket of pus. So some bacteria, like TB, tuberculosis bacilli, actually resist digestion by macrophages. So they actually basically remain inside of the body alive, and they form little tumor-like growths. So it's not really a tumor. Instead, it's like little pockets that are consisting of these tuberculosis bacilli, these little bacterial cells. We call those granulomas because they're little pockets of granules. So the infected area is going to be totally surrounded by macrophages and form kind of like a capsule. And the bacteria may remain inactive forever, or if something happens and the person's immunity goes down, and they don't have that kind of protection around the granuloma, the bacteria may break free and actually cause the disease. That's one reason some people are carriers of TB but may not actually be infected and have the actual disease. Antimicrobial proteins will also enhance the innate immune system defense. They can sometimes attack the microorganism like a bacteria directly, or they block its ability to reproduce and make more, and it lets other parts of the immune system kind of catch up. Some really important antimicrobial proteins are called interferons. We also have complement proteins. So interferons are a special kind of immune protein. And cells infected with viruses actually typically secrete interferons to basically warn healthy neighbors that there's a problem. They do a lot of other things, but if you remember, we talked about virus and virus-infected cells being killed by NK cells. So interferons can be very important for triggering the NK cells to kind of attack a cell because of maybe a virus or a cancer. So we won't really talk about more details other than just want you to recognize that an interferon or the different types of interferon proteins are really important for warning that there may be a potential infection or a problem like a virus or a cancer. And so here's actually a picture, a diagram showing kind of how interferons work. You're welcome to look at on your own time. And then lastly, as we mentioned, we have complement. So the complement is actually a system of multiple blood proteins that circulate and actually can bind together and provide a major mechanism for getting rid of foreign substances, particularly destroying bacteria. They enhance both the innate and the adaptive immune systems. And there's several different pathways. I do not, for this class, expect you to know the different pathways. You may have to learn them later on. But 3 different ways, essentially, that we use these complement proteins to get rid of bacteria. So one of them actually uses antibodies to surround the foreign substance. We'll talk about antibodies more next week as they come from the B lymphocytes, the B cells. And that helps to actually get things prepared for the complement proteins to start breaking down the bacteria. We'll kind of skip over that because, like I said, I don't expect you to know the distinct differences between complement. But each pathway in complement involves activation of proteins and a very specific orderly sequence. So the first step catalyzes or helps to make ready the next step. And it keeps happening in a very specific sequence, a very specific order. All the different pathways converge on the same proteins, the same step in the pathway. And that basically will cause inflammation, increased phagocytes in the area to come around to gobble up the foreign substance, and actual cell lysis. So the lysis meaning cut, so cutting of the bacterial cell. So cell lysis depends on something we call the MAC complex, the membrane attack complex. And you can imagine it's almost like it pokes little holes inside of the bacteria. And what happens is, kind of like if we poke little holes in our skin, we start leaking things out and bad stuff can make it in. Same idea. The MAC complex at the end of complement basically pokes holes into the bacteria so some stuff that the cell needs to survive leak out into the surrounding area. Some things that could potentially harm or make it harder for the cell to survive make it inside of the cell. So it basically kills the bacterial cell. And so here's actually a picture, a diagram of what it looks like. And the MAC attack complex that forms, it's going to poke holes into the bacterial cell. And that last big thing we talked about that's part of the system of the second line of defense is actually a fever. So an abnormally high body temperature that is systemic in response to invading microorganisms is called a fever. Basically, leukocytes and macrophages are exposed to foreign substances that secrete what's called pyrogens. Okay, pyro is the Greek word for fire, so literally fire causing things. It makes the temperature go up. So if you hear the word pyrogen, it's just something that causes your temperature to increase in the body. It affects the hypothalamus, causing the temperature to rise. And a moderate fever can actually be a good thing. You don't want it to get too extreme, but a moderate fever actually causes the liver and the spleen to sequester iron and zinc, which most microorganisms need to survive, so it kind of prevents them from getting those nutrients. And it also helps to speed up the metabolic activity, which can increase a lot of things that are going on with repair. And here's kind of a summary of those different aspects of the second line of defense. All right, and we'll pick up next week talking about the details of the adaptive immune system.